Our fourth strategy is genetics counseling and intervention. Interventions based upon genetics have been part of medical and public health practice since at least the 1960s, when it was recognized that abnormalities of single genes for such conditions such as Tay-Sachs disease found among certain Jewish communities and sickle cell anemia found among African Americans could be detected by testing, and potential parents could be counseled on the risks associated with childbearing. It was also recognized that chromosomal abnormalities that produce Down syndrome could be detected at an early stage in pregnancy. In addition, certain genetic defects, such as PKU, could be recognized at birth, and in the case of PKU, relatively simple dietary interventions could prevent the severe retardation of mental development that would otherwise occur. Genetic prevention looks at a single gene or gene combination before birth. This approach incorporates efforts to prevent the occurrence of a single gene or multiple gene combinations that are likely to produce disease. Knowledge of the human genome holds promise for expanding this approach beyond diseases caused by a single gene defect to diseases that depend upon multiple genes. Additional approaches to genetic counseling and intervention include genetic detection. This is identifying genetic defects and implementing early identification and intervention after birth. The efforts are aimed at the detection of genetic defects with intervention to prevent expression of genes. Also, gene environment protection. This is genetic testing that holds out the possibility of defining combinations of genes that identify individuals who are especially likely to develop disease when they experience specific environmental exposures. This is identifying the gene-environment interaction that may lead to identifying those who are at high risk if they work in certain occupational situations. Genotypic-based screening for early detection looks at combinations of genes that may identify groups that are at high risk of common diseases that can be targeted for screening. For instance, studies suggest that certain common cancers, such as breast, prostate, and colon, have genetic factors associated with them in 30 to 40% of the disease cases. This is looking for a predisposing genetic pattern to identify those who might need more vigilant screening. An example of two high-profile individuals who used this type of screening are Christina Applegate and Angelina Jolie. Both had the genetic mutations that signify high risk for breast cancer, and so both elected to have prophylactic mastectomies. Our fifth strategy to address the burden of communicable diseases is research. Scientists research treatments to find out what are effective and what is not effective. But this costs money. A lot of our tax dollars are spent supporting the National Institutes of Health, who give money to a lot of universities and other organizations to conduct research. However, if a disease does not have a large burden on the community, tax dollars are not spent. And so we have what are called orphan diseases. These are conditions that have low incidence rates, and so we spend little money on researching treatments until celebrities get them, and then they raise a lot of money. So, for example, Lance Armstrong had prostate cancer, which brought a lot of awareness to that disease. Michael J. Fox developed Parkinson's disease and has brought a lot of awareness and raised a lot of money for research into that disease. Christopher Reeves, who's no longer with us, but you may remember him if you've seen the old Superman movies, fell off a horse and had a spinal cord injury. He raised a lot of money for stem cell research because he believed that that would be a treatment to treat spinal cord injuries.